Good morning, everyone. Myself, sneha suman purbia from Group 316. Here is my topic, the structure of a membrane. We know that the cell membrane, that is also known as a plasma membrane, is a double layer structure which is made up of lipids and proteins. This that surrounds by a cell that is separated from the cytoplasm from its surrounding environment. This cell membrane is a selectively permeable, means which can control the amount of substance which can come in and go out. All cells have the cell membranes. This is the structure of a cell membrane. See, uh, these is the lipids and this is the lipid, so we can say that it is a lipid bilayer structure. It contains the proteins which are embedded in inside the lipids. These are the some are the integral proteins, some are the extra integral some are integral proteins, some are external proteins. On the proteins we have a carbohydrate chains which are present on the this cell membrane. um, you can see this is the these carbohydrates are present outside the cell, and in the inside the cell, the cytoplasm, where the cytoplasm is present, we have some protein channels. See, a biological membrane, it is a dynamical organized system. It is necessary to study both the structure and the dynamic of its behavior of the cell membrane. Biological membrane consists mainly of a lipids and protein, as I say. The cell of the mammals also contains small amount of carbohydrate that is associated with the proteins that are mostly glycoproteins, and with lipids. The inner membrane largely contains phospholipids, and the plasma membrane also contains the neutral lipids also. Examples, if we are talking about the erythrocytes, RBCs, in the RBCs the 30% of lipid is present in the form of cholesterol. In most of the cases, membranes are heterogeneous. They contain whole families of phospholipids and the lipids. For example, the membrane of human erythrocytes contain no less than 20 types of lecithins. Lip, lipids are built up of a polar and non-polar structure. Polar what? Polar head and two non-polar carbohydrate tail. See in this diagram, we these lipids having a polar head and non-polar tail. Polar head and non-polar tails, which possesses hydrophobic properties. As far back as 1935, Daniel and Davison proposed a theoretical model of composition of a biological membrane that known as a unit model. The unit model is considered to be a bilayer of mixed polar lipids and their hydrophobic tail extending inwards, and their hydrophobic heads exposed on the surface, where they are bound to the outer monomolecular protein layer. The main source of information about the cell structure and the cell membrane is by electron microscopy. Another is the main idea of organization of a unit membrane. The bilayer arrangement of a lipid is correct. It is proved by the X-ray data, as you see in the picture. As regard their protein code, the arrangement of a proteins differ from the suggest in symmetrical unit membrane models. According to their which membrane proteins are hydrophilic surface which become bound to the polar end of the lipids. Isolated membrane proteins and the membrane have also been studied by indirect methods by the action of proteolytic enzymes on them. By incorruption of various levels into a membrane protein, it has been found that the membrane proteins can be subdivided into two classes. Some of the proteins are bound only the membrane surface, that are the globular proteins which functions in an aqueous environment. They have hydrophilic surface. The protein of a second class are capable of penetrating the membrane and uh, interacting with the hydrophobic tails of the lipid. These proteins are insoluble in water, their surface being hydrophobic. The investigation of the membrane carried out by the means of uh, infraspectroscopy, spec- spectropolarimetry, or NMR, etc. The point of the diversity of the protein structure are to interprotein interaction, which are not taken into account into a unit membrane model. It has been established that the proteins are arranged in asymmetrically in the membrane range. There are several modifications of the unit membrane model. At present, the model that is thought to be closer to the truth is the fluid mosaic model of the cell membrane that is shown in this diagram, or you can see in this diagram also. This model to the bilayer of the lipid. As a matter of the fact, the artificial lipid membranes with the bilayer structures, they are found to be similar to the biological membrane in many aspects. Artificial membranes are produced when the mixture of the phospholipid and the neutral lipids dissolved in an organic solvent is allowed to contact with water. In this way, black membrane are produced, that is thin layer 10 nanometer thick, which is devoted to interface color. It has been found that these membranes are have the bilayer structure indicated. Compared to the properties of the artificial bilayer uh, lipid membrane, with those biological membrane, artificial membrane display no metabolic activities and are not so highly sensitive as biological membrane, but they stimulate the important properties of biomembrane and enable the study the transport of of material and excitability. It has been established that the various complex liquid crystal structures of a long range order are formed in lipid water system. This system are the scientific interest in own rate. There is ground for behaving that the multiphases natural of a lipid system is directly associated with the function of a biological membrane. So, this is all about my topic, the cell membrane. Thank you.